In 2014, a team of geneticists extracted DNA from elongated skulls found in Paracas, Peru. The skulls were over 3,000 years old and had cranial capacities 25% larger than modern humans. The initial DNA analysis showed genetic markers that didn't match any known human population. The lead researcher said the results were so unusual that the sample must have been contaminated. They ran the test again, same results. Then they tested 5 more skulls. Every single one showed the same impossible genetic anomalies. The skulls have been sitting in museums for decades, while archaeologists insisted they were just normal human skulls deformed through cranial binding. The DNA says otherwise. Here's what scientists actually found when they tested the genetics of these ancient elongated skulls, and why the results challenge everything we thought we knew about ancient Peru. The Paracas skulls were discovered in 1928 by Peruvian archaeologist Julio Tello in the Paracas Peninsula of southern Peru. He found over 300 skulls in elaborate tombs dating back to approximately 800 BCE. What made them extraordinary wasn't just their elongated shape, but their structural differences from normal human skulls. Here's what makes them anatomically strange, which was practiced by many ancient cultures, deforms the skull's shape, but doesn't change its volume. You're reshaping the same amount of bone. The Paracas skulls have 25% greater cranial capacity than average human skulls. Some specimens measure up to 1,500 cubic centimeters compared to the modern human average of 1,200 cubic centimeters. But here's what's even stranger. The skulls have only one parietal plate instead of 2. Normal human skulls have 2 parietal bones that fuse together at the sagittal suture running down the center of the skull. Some Paracas skulls lack this suture entirely and appear to be formed from a single bone. This isn't deformation. This is a completely different skull structure. Physical anthropologist Brian Forster, who has studied these skulls extensively, the bone density is also different, being both thicker and heavier than typical human cranial bone. These aren't features you can create through binding infant heads. The traditional explanation never made sense. Archaeologists claimed that the Paracas culture practiced extreme cranial binding from infancy, wrapping babies' heads with boards and cloth to create elongated shapes. But cranial binding deforms shape. It doesn't increase volume, change fundamental bone structure, or create single-plate parietal bones. In 2014, geneticist Melba Ketchum and her team extracted DNA from tissue samples taken from the interior of one Paracas skull. The sample was taken from the skull's interior to minimize contamination from handling. Standard DNA sequencing protocols were followed. What came back was impossible. The mitochondrial DNA showed genetic markers that didn't align with any known Native American haplogroups. Mitochondrial DNA is inherited exclusively from the mother and is used to trace maternal lineage. Native American populations fall into specific haplogroups labeled A, B, 4D, and X. The Paracas skull DNA showed markers that didn't fit any of these categories. But here's where it gets terrifying. The DNA showed some markers consistent with European and Middle Eastern populations. This was in skulls that were carbon dated to 800 BCE, over 2,500 years before European contact with Peru. The genetic signature suggested ancestry from populations that shouldn't have been in South America at that time. The initial response from the scientific community was predictable. Contamination. The sample must have been contaminated by modern handling or by European DNA from later burials in the same area. So they tested again with stricter protocols. Same results. Then they tested 5 additional skulls. Every single one showed the same genetic anomalies, mitochondrial haplogroups that didn't match known Native American populations, nuclear DNA that suggested mixed ancestry from geographically impossible sources for the time period. Here's what the lead researchers said. The DNA was too degraded to get complete sequences, which is normal for 3,000-year-old samples. But the partial sequences they did get were consistent across multiple samples. This wasn't contamination. This was the actual genetic material of whoever these people were. One specific finding from the DNA analysis caused significant controversy. Several skulls showed mitochondrial haplogroup U, specifically U2E. This haplogroup is found in modern populations from Europe, the Middle East, and parts of Asia. It's not found in Native American populations. It's never been found in pre-Columbian South American remains, but it was in the Paracas skulls. Multiple samples from different individuals, all showing the same impossible haplogroup. Carbon dating confirmed the skulls were from 800–100 BCE. That's 2,000 years before any documented European contact with Peru. The explanations fall into three categories. First, contamination. Despite multiple tests with strict protocols, maybe modern DNA contaminated the samples. Second, unknown migration. Maybe populations from Europe or the Middle East reached South America thousands of years earlier than historical records indicate. Third, something else entirely that we don't understand. But here's what makes contamination unlikely. The samples were taken from the interior of skulls that had been buried for 3,000 years. The DNA was highly degraded, which is exactly what you'd expect from ancient samples. Modern contaminating DNA would be less degraded and easy to identify. The sequences they found were degraded in patterns consistent with ancient DNA, not modern contamination. The unknown migration theory has problems too. There's zero archaeological evidence of European or Middle Eastern populations in South America before Spanish colonization. No artifacts, no trade goods, no linguistic influence, just these genetic markers in skulls with impossible anatomy. Beyond the DNA, the physical anthropology of these skulls presents problems that haven't been adequately addressed. Dr. Michael Alday, a maxillofacial surgeon who examined several specimens, noted features that artificial cranial deformation cannot produce. First is the cranial volume increase. Binding an infant's head changes shape by redistributing the same volume of bone and brain tissue. It doesn't create more volume. The Paracas skulls have measurably larger cranial capacities. Where did the extra volume come from? Second is the single parietal plate. Some specimens show no sagittal suture at all. The entire top of the skull appears to be formed from a single bone. Human skulls develop from separate bones that fuse together during growth. This fusion happens in predictable patterns. A skull with no parietal division isn't just unusual. It suggests different developmental biology. Third is the bone density and weight. The skulls are significantly heavier than normal human skulls of equivalent size. The bone itself appears denser. Cranial binding doesn't change bone density. It shouldn't add weight. Fourth is the foramen magnum position. The foramen magnum is the opening at the base of the skull where the spinal cord enters. In the elongated skulls, this opening is positioned differently than in normal human skulls, suggesting different biomechanics of how the head attached to the spine. Here's what medical experts keep saying. These features can't all be explained by cranial binding. You can deform shape. You can't increase volume, eliminate bone sutures, change bone density, and reposition the foramen magnum through external pressure on a growing skull. Either these are a completely different human subspecies that we don't have any other evidence for, or the DNA results and anatomical anomalies are telling us something that doesn't fit our understanding of ancient Peru. The Paracas culture treated these elongated skull individuals differently from normal burials. The tombs where the skulls were found contained elaborate grave goods, textiles of exceptional quality, and evidence of special status. These weren't normal people. They were treated as elite or separate. The textiles found in the tombs are themselves anomalous. The Paracas culture produced some of the finest textiles in the ancient world, with thread counts that rival modern fabric and dyes that have remained vibrant for 3,000 years. The techniques used to create these textiles are still not fully understood. Some patterns show knowledge of complex geometry and mathematical relationships. But here's what's particularly interesting. The elongated skull burials were separate from normal human burials. They were placed in distinct tomb complexes with different architectural features. The burial positions were different. The grave goods were different. The culture was treating these individuals as fundamentally different from the general population. Some researchers have suggested this indicates a ruling class that practiced cranial binding as a status marker. But if that were true, we'd expect to see some skulls with partially successful binding, some with failed attempts, some variation in the degree of elongation. Instead, we see consistent anatomical features across hundreds of specimens. The most disturbing possibility is that these weren't artificially modified humans at all. They were a distinct population group that lived in ancient Peru, interbred with the local population to some degree based on the DNA evidence, and then disappeared from the historical record entirely. Elongated skulls aren't unique to Peru. They've been found in Egypt, Malta, Bolivia, Mexico, and even parts of Europe. The geographical distribution is global, spanning cultures that had no known contact with each other. The traditional explanation is independent invention. Multiple cultures independently developed cranial binding practices for aesthetic or status reasons. This is possible. Many technologies were independently invented in different parts of the world. But here's what's strange about that explanation. The skulls from different continents show remarkably similar anatomical features beyond just elongation. The size increase, the bone density changes, the parietal plate anomalies appear in specimens from Peru, Egypt, and Malta. If these were independent cultural practices of skull binding, why would the results be so anatomically similar? Some of the Egyptian elongated skulls, particularly those from the Amarna period during Akhenaten's reign, show the same cranial capacity increases and structural anomalies as the Paracas skulls. They're on opposite sides of the world. They're from different time periods, yet the physical anthropology is remarkably similar. This suggests either a shared technique that was transmitted across vast distances through unknown contact, or a shared genetic basis that existed in populations across multiple continents. Neither explanation fits comfortably with current archaeological consensus. Since the initial 2014 DNA results, several additional genetic studies have been conducted on elongated skulls from various locations. The results haven't clarified the mystery. They've deepened it. A 2018 study published in PLOS One analyzed DNA from 14 individuals buried at the Tiwanaku site in Bolivia. Several had elongated skulls. The DNA showed they came from multiple distant populations, including some with ancestry from populations separated by thousands of miles. The study concluded that elongated skull individuals were cosmopolitan and mobile, contradicting the idea that they were isolated ruling elites. But here's what the study couldn't explain. How did individuals with genetic ancestry from such distant and varied populations end up in the same burial site in ancient Bolivia? The DNA showed ancestry from regions including the Amazon, the Andes, and areas far outside South America. This was 1,500 years ago, long before known long-distance travel capabilities. A separate genetic analysis of Egyptian elongated skulls found similar geographic inconsistencies. Genetic markers suggested ancestry from populations that shouldn't have been in Egypt at the time of the burials. The researchers concluded that either there was far more migration and population mixing in the ancient world than we currently understand, or the genetic results are being contaminated in ways we can't detect. The pattern across studies is consistent. Elongated skulls show genetic markers that don't match the expected ancestry of the location and time period where they're found. Either ancient migration was far more extensive than archaeological evidence suggests, or something else is producing these genetic anomalies. Theory 1 is artificial cranial deformation. This is the consensus archaeological view. Ancient cultures bound infant skulls to create elongated shapes as status markers. The anatomical anomalies are either exaggerated or the result of unusual binding techniques we don't fully understand. The DNA anomalies are contamination or ancient migration we haven't documented. The problem with this theory is it requires ignoring multiple lines of evidence. The cranial volume increases that binding can't produce. The structural changes that binding can't create. The consistent DNA anomalies across multiple tested specimens from different locations. Theory 2 is unknown ancient migration. Populations from Europe, the Middle East, or other distant regions reach South America thousands of years before documented contact. They interbred with local populations, creating the genetic signatures we see in the DNA. They also brought knowledge of skull modification techniques that spread globally. The problem here is complete absence of supporting evidence. No European or Middle Eastern artifacts in pre-Columbian South America. No linguistic influence, no cultural markers, just the DNA in these specific skulls. Theory 3 is a lost subspecies of human. The elongated skulls represent a distinct human population, possibly a subspecies that had different cranial anatomy and lived alongside Homo sapiens. They interbred with modern humans, which explains the mixed DNA results, and then went extinct or were absorbed into the broader population. The problem with this theory is that it requires a subspecies we have no other evidence for. No separate fossil record, no distinct archaeological culture, no documented history. Just these skulls and their anomalous genetics. Theory 4 is the one nobody wants to say out loud. These weren't human at all, or they were human hybrid of some kind we don't understand. The DNA shows markers we can't classify. The anatomy shows structures that don't match known human variation. Maybe they were something else entirely. This theory violates everything we know about human evolution and has zero supporting evidence beyond these anomalous skulls. But it's the theory that keeps appearing in fringe research because none of the conventional theories adequately explain all the evidence. Here's what makes this genuinely terrifying. We have physical remains with impossible genetics and anatomical features that shouldn't exist. Multiple labs have confirmed the DNA anomalies. Medical experts have documented the structural differences. Yet the scientific consensus is that this must be some combination of contamination, unknown migration, and unusual binding techniques. That's not scientific. That's assumption-based reasoning, designed to fit uncomfortable evidence into existing frameworks. If the DNA is contaminated, it's contaminated in consistent ways across multiple specimens from different sites, tested by different labs using different protocols. If the anatomy is explainable by binding, we should be able to replicate those results experimentally. We can't. The truth is, we don't know what these skulls represent. They could be evidence of ancient migration patterns we haven't discovered. They could be genetic anomalies from isolated populations. They could be something we don't have a framework to understand yet. What's certain is that dismissing the evidence because it doesn't fit current models isn't science. The DNA results are real. The anatomical anomalies are documented. The burial patterns are distinct. Something about these individuals was different enough that ancient cultures treated them as separate and special. Whether that difference was cultural, genetic, or something else entirely, the skulls are sitting in storage facilities and museums right now with genetic material that doesn't match any known human population and anatomy that doesn't match normal human skulls. That's not a solved mystery. That's a mystery we've decided to stop investigating because the answers might be uncomfortable. The elongated skulls of Paracas aren't evidence of aliens or ancient astronauts, but they might be evidence that human history, migration, and genetic diversity in the ancient world were far stranger and more complex than our current models allow for. And that possibility might be more terrifying than any supernatural explanation.